Hi, I would like to thank to Norberto. Gracias por invitarme. And also I would like to thank to the organization staff for the hard work that they are doing. As Mark has said, I'm going to talk about my studies on neuroanatomy. First of all, I would like to do a brief introduction about the point of view of a neuroanatomy laboratory, because we have to consider that the cerebral cortex is an extremely specialized structure with a high complexity organization, and to have an idea of the kind of study that we are going to, I am going to expose here, in one single cubic millimeter of cerebral cortex tissue, we have about 25,000 neurons and 1,000 million of synapses. Basically, I am going to talk about the elements of the cerebral cortex, and I would like to introduce the main types of neurons. I don't know if you can see the pointer. No. Okay. These kind of neurons were described by Santiago Ramón y Cajal, and there are mainly two types of neurons, which are the pyramidal neurons, as we can see here in this part of on the right of the slide. Thank you. Yeah, thank you very much. In this original draw made by Cajal, these kind of neurons were already described, and this is a picture taken from an original preparation made by Cajal using the Golgi method, in which we can distinguish the pyramidal neuron, which also the Cajal named as long axon neurons, because their axon projects in areas outside the area in which the neuron is located, and also the interneurons. Cajal named these neurons also short axon neurons, because the axon is located in a local area. And the pyramidal neurons are also considered the projections neurons of the neocortex. They use glutamate as a neurotransmitter and are considered to be excitatory, and they are about 70 or 85% of the total population of neurons, depending on the region and the kind of cortex that we are studying. And the pyramidal cells can be classified according to the projection sites. And the interneurons are considered to be inhibitory because they use GABA as a neurotransmitter. They represent about 15–30% of the overall population. And they are morphologically and neurochemically very heterogeneous. Under the point of view of the epilepsy study, it's very important to keep in mind that the corticocortical connections are provided by pyramidal neurons, because they are the main projection cells, and they provide more of all excitatory synapses in the cortex, and they are responsible for propagation of cortical activity through their axon collaterals. And more than 80% of all excitatory synapses in the cerebral cortex are originated from pyramidal neurons. And the neurons, as elements, are connected by cortical synapses, and these synapses, this contact, can be morphologically identified, and there are two main types of cortical synapses in the mammal's brain. As we can see here, there are the asymmetric synapses, and they are characterized because they have a very thick postsynaptic density, and they use glutamate as neurotransmitter, and they are considered to be excitatory, and they are about 70–80% of the total synaptic contacts in the brain. And as we can see here, when we use some labeling for the excitatory markers, are the vesicular glutamate transporter, they are labeling, whereas the symmetric synapses are characterized because they have a very thin postsynaptic density, and they can be labeled using inhibitory markers like the GABA or the synthesizing enzyme, and they are inhibitory, and they constitute about 15% of the total population of synapses. And it's very important to study the proportion of these two kind of synapses, because changes or differences in the density and in the proportion of asymmetric and symmetric synapses would represent modification of the excitatory and inhibitory balance of the cortical circuits. So if we have a complete idea of the elements and how they are connected, we have to consider that the cortical circuits are strictly regulated, and any alteration may produce changes in the information processing and then in the cortical function. And we can consider that epilepsy is a consequence of a malfunction of these cortical circuits. And our studies on epilepsy are focused in the temporal lobe epilepsy, which is the very common form of epilepsy. And in most of the cases, we found hippocampal sclerosis as in the resected mesial temporal tissue. And in about 30% of the TLA patients with pharmacoresistant epilepsy, the surgery is an option to reduce the number of seizures. In our laboratory, we work with an epilepsy surgery unit in the Hospital de la Princesa in Madrid. Basically, the neurosurgeon opened the school. He exposed the surface of the brain, and he puts intraoperative recording in which he's able to record every point of the surface of the cortex. And this is an example of any patient. And also sometimes he puts a mesial electrode inside the mesial structure in the amygdala and hippocampus. And after delimiting the active area, the neurosurgeon removes specifically the part of the tissue which is supposed to be active, and we can fix this part of the tissue in our laboratory to perform the histopathological studies. Basically, the hippocampal sclerosis is characterized by a gliosis and neuronal loss, which is very prominent in the CA1 region. I can see clearly. In the CA1 region, followed by CA3 and CA2. And in the dental gyrus, we can find also alterations. As we can see here, there is a specific staining to label neurons, and we can observe clearly the neuronal loss in this area, and also the alterations in the structure of the granular layer of the dental gyrus. And as we can see here, the neuronal loss and gliosis is different in every hippocampal subfield, and sometimes where we find that in the regions with massive neuronal loss, there is an abundant glial population. As we can see in this nasal stained section, these small dots in blue are glial cells, whereas in other regions, as subiculum, the aspect is normal, and there are no pathological alteration in an optical examination. And also in the dental gyrus, when we examine different patients, we can find that there is a variety of changes in the alteration of the dental gyrus. This is the control patient without sclerosis, and these are 3 different patients with hippocampal sclerosis. And we can observe that there are several different changes in which there is a typical dispersion of the dental gyrus cells, and also we can find a double layer or clustering. So we think that the hippocampal sclerosis is not a single pathology, and the neuronal loss and gliosis vary between different hippocampal regions and patients. The variability of these alterations makes difficult to determine which are inducing the epileptic activity. The epileptic activity itself may produce similar histopathological alteration than hippocampal sclerosis, and it has been reported that in experimental models, chemically induced or electrophysiologically induced, we can observe the same histopathological alterations. And also it's important to keep in mind that in some human non-epileptic patients which suffer a toxicity, we can find the same alterations. And also I would like to show you this experimental model, which is a chemically induced seizure model made in monkeys, in which it was made an injection of alumina gel into the amygdala, and after 15 days of the injection, the monkey suffered seizures. When the tissue was analyzed, the alumina injection site showed the same alterations in the dental gyrus that in the human hippocampal sclerosis. Also, another point of very important to keep in mind when we study the hippocampal sclerosis is that some patients with severe alterations in the hippocampal formation do not develop epilepsy. It was shown by the study on autopsies because these authors revealed that there was a severe damage in the hippocampal formation in non-epileptic patients. So in our study of the pathology, we have two main questions, and they are: which alterations are necessary in the neuronal circuitry to create an epileptogenic region, and why some patients can be well controlled on anti-epileptic drugs, whereas others not? We have to consider that the hippocampal formation is involved in a very complex circuit, in which, if we consider that the hippocampal sclerosis is the origin of the epileptic activity, and we know the connectivity of the hippocampal formation, we have to keep in mind that the CA1 pyramidal cells project predominantly to the subiculum, and the subiculum is the origin of the major subcortical projections, as well as the projections to the entorhinal cortex. And previous studies have shown that there is an epileptiform activity in the subiculum CA1 region, as we can see here in this slide. And this group reported epileptiform activity in this specific region, but we would like to know what happened with these synaptic circuits in this particular region. So we perform a specific detailed ultrastructural analysis in this region of the sclerotic and non-sclerotic hippocampus. And we consider to make our studies in the pyramidal cell layer in the CA1 region, because it suffers a severe neuronal loss in the sclerotic hippocampus, in the transitional region, and also in the subiculum, because the sclerotic subiculum is apparently non-altered. And what we found is that in the sclerotic CA1, we could observe numerous glial processes and a dramatic loss of synaptic contacts when compared to the non-sclerotic tissue. In the transitional region, we also studied the neuropil and the synaptic contacts there. We observed a moderate number of synaptic contacts, and in the subiculum from the sclerotic hippocampus, we found a normal neuropil, which was almost undistinguishable of the non-sclerotic subiculum, and no ultrastructural alterations were found. We decided to quantify this alteration using an estimation of the synaptic density, and for this purpose we made an estimation using the size frequency method, and we basically count all the synapses in the pictures. Also, we could distinguish between the asymmetric and the symmetric synapse, and so we had a good estimation of the cortical circuits in this region, about the imbalance of the excitatory and inhibitory. And what we obtained is summarized here. There is a significant decrease in the synaptic density in the sclerotic CA1. As we can see in this graph, there is a dramatic reduction in the general number of synapses in the sclerotic CA1, but in the sclerotic subiculum, the number of synapses was almost similar to the non-sclerotic. We also consider, as is found here in this table, we also consider the differences between the asymmetric and symmetric synapses, because they are the excitatory and inhibitory and the total number of synapses. And when we compare the subiculum from non-sclerotic and the sclerotic, we couldn't find any difference. However, we found that when we compare the different regions inside the sclerotic subiculum, we found a gradual decrease in the synaptic density in function of the degree of histological alteration. As we can see in this graph here, there is a gradual decrease in the number of synapses according to the neuronal loss and the gliosis that is shown in these regions. So, if we put together all this information, we have to consider that the hippocampus is integrated in a very complex circuit, and in the hippocampal sclerosis, there is a massive neuronal loss, and probably this part of the circuit is altered. And it's possible that the neurons located here, which are lost, are not connected anymore to the subiculum. So, if we consider this fact, we think that the subiculum may serve as an hyper-excitable fringe area which would amplify the relatively few excitatory outputs from the sclerotic Hammond horn. So, as a summary of this part, the sclerotic CA1 shows very few synapses, making it unlikely that this region contributes to epileptiform activity, and therefore epileptiform activity might arise from other hippocampal regions. And the synaptic density in the transitional region contains a number of synapses similar to that in the subiculum, which may arise from the surviving CA1 neurons and from other hippocampal and extrahippocampal afferent systems. And the sclerotic subiculum shows abundant synapses, similarly to non-sclerotic hippocampus. And we propose that the subiculum is a critical region in epileptogenicity in the hippocampal sclerosis. And the second part of my results are focused in the study of the alteration in the microvascular network in the sclerotic hippocampus. And this alteration in the vascular system has been widely studied previously, and it was reported an increase in the density of blood vessels in both sclerotic hippocampus of patients with TLA and animal models. And it has been previously reported in several studies that there was an increase in the number of blood vessels, as we can see here, in the sclerotic CA1 compared in the normal tissue. We did a specific study of the blood vessels, and we used the alkaline phosphatase enzyme, which is expressed in endothelial cells of several blood vessels, and this enzyme is located in all the regions of the brain, and also it has been proposed to be involved in the GABAergic neurotransmission. When we use this staining in our tissue, in the normal human hippocampus, we can see the staining is distributed in all the regions. And in this slide, I did a counter-stain section staining with NISL, and we can see all the fields here with the neurons. And when we study in details this region, we can see that we can see clearly that the blood vessels stain are homogeneously distributed in all the fields. However, when we did the same study in the sclerotic hippocampus, we observed a decrease in the staining in several regions, and in a detailed picture in a higher magnification, we saw that there is a severe loss in the APA staining in the sclerotic CA1, which was concurrent with the neuronal loss and gliosis. As we can see here, in the upper part of the slide, there is this sclerotic CA1, and we can observe very few AP-positive blood vessels and a lot of glial cells. However, in the normal CA1, we observe a lot of AP-positive blood vessels, and these labeled neurons were abundant. So we quantify the AP-positive blood vessels in the sclerotic tissue, in the non-sclerotic tissue, and in the autopsy tissue, in the different regions of the hippocampus, and we found that there is a significant reduction in the density of AP-positive blood vessels in the CA1 region from sclerotic hippocampus compared to autopsy, about 60 or 70%, and compared to non-sclerotic patients. So we also wanted to know what happened with the other previous reported increase in the blood vessel. And when we used a specific staining of collagen 4, which is a component of the basal lamina, we found that when we quantified the staining, we found an increase, about 160% in the staining in the blood vessels in the sclerotic hippocampus. So we found a contradiction between the lack of staining in the AP-positive blood vessels and the increase in the collagen 4 positive blood vessels. And when we study these blood vessels in a higher magnification, this is the control tissue here, and this is the sclerotic tissue, and we could find that there were alterations in the blood vessels, in the stained blood vessels, and we found an abnormal surface in the blood vessels like protrusion, and we also find positive structures like reticular structures, and we couldn't be sure if there were blood vessels or not. So we examined this part of the sclerotic tissue. These alterations were found in the sclerotic CA1, mostly in those regions in which there were the massive neuronal loss and gliosis. And as we can see here at higher magnification, we found that there were a lot of elements positive for the collagen 4, but they were as abnormal structure. They were not like blood vessels. So we decided to investigate the structure of these elements under the EM, and also we considered to examine under a detailed examination in the light microscopy. We estimate how many of these blood vessels were present in the non-sclerotic and in the sclerotic tissue, because we have the contradiction between the AP staining and the collagen 4 staining. We perform a method which is useful to count the volume fraction occupied by blood vessels. We found that in the non-sclerotic CA1, there were a lot of blood vessels, and in the sclerotic CA1, there was a decrease in the blood vessel, in the volume fraction of blood vessels. And when we quantify this data, we saw that there is a 60% of decrease, which was very concurrent with the AP staining. And since we have the staining in the collagen 4 in the sclerotic CA1, we decided to go to the EM to study the ultrastructure of these blood vessels. And when we compare the non-sclerotic and the sclerotic CA1, as we can see here, higher magnification, in the normal tissue, the surface of the blood vessels are smooth, and we found the endothelial cells, normal structure. But when we examine the sclerotic CA1, we found a very strange structure in the blood vessels, like protrusions and a kind of abnormal structures. And also when we examined the lumen of these blood vessels, we found that it was virtually full of kind of glial processes or fibers. So we conclude that there is a consistent loss of normal blood vessels in the human sclerotic hippocampus, and the alteration of blood vessels can be considered as another hallmark of human hippocampal sclerosis. But the significance of this blood vessels reduction in the hippocampus is not clear. And as a summary of both parts of my talk, the sclerotic CA1 shows neuronal loss, very few synapses, and a consistent loss of normal blood vessels. And all these changes make it unlikely that CA1 region contributes to epileptiform activity. Therefore, epileptiform activity might arise from other hippocampal regions. And the significance of blood vessel alterations in the hippocampus is not clear in terms of epileptogenicity. Also, the synapses in the transitional subiculum CA1 region and in the subiculum in the sclerotic hippocampus may arise from the surviving CA1 neurons and or from other hippocampal and extrahippocampal afferent systems. Finally, I would like to mention that our future studies are focused on the detailed 3D ultrastructural analysis of the pathologic tissue. For this purpose, we now have a new microscope. This is a kind of focused ion beam scanning electron microscope, which allows us to obtain similar pictures to those obtained with the transmission electron microscopy, but we have the advantage that we obtain serial images in a fully automatic way, and we eliminate the serial section in collecting an ultrasound section, and we avoid also the problems of the technical approach. And we can obtain serial reconstructions of large volumes of tissue, and we can study any part of the tissue that we have. This is very useful for us because in the study of the synaptic contacts, we can choose the synaptic that we want to study, and using a specific software, as we can see here in this movie, we can study the tissue in the different planes because it's a serial stack, And also we can reconstruct the synapse or the structure in which we are interested. Also we can obtain the data on every single synapse or synaptic contact here. And as we can see in this picture, we can distinguish perfectly which synapses are excitatory and inhibitory. In green, we have the excitatory, and in red, we have the inhibitory. And also this software provides a lot of data of this synapse, like the size or the position or useful information to determine which is the state of the cortical circuits. And this part of the information about the synaptic apposition surface is very important in terms of the neuroanatomy, because the size of the active zone, which is here, is proportional to the number of docketed synaptic vesicles and to the probability of the vesicles release. And the released neurotransmitter molecules interact with the postsynaptic receptors present in the postsynaptic density. And the area of the PSD is proportional to the number of postsynaptic receptors. So the size and the shape of the active zone in the presynaptic terminal and of the PSD provide useful data in terms of synaptic function. So now we would like to investigate these structures in the pathological tissue to obtain that data on the synaptic connectivity. And also, I would like to show you my last movie. As we can see here, there is a block of human brain tissue, and it's taken from the cerebral cortex, in which we have fully reconstructed all the synaptic contacts inside. And as we can see here, they are turning in the yellow color because the apposition surface is obtained by an algorithm, and we can study every one individually, and we can obtain all the data on the synaptic size and also about the apposition surface, providing the information to study the synaptic circuits. And thank you for your attention.